Good day, everyone. Today, we will be talking about manual blood cell counts, specifically erythrocyte and leukocyte counts. Let's first take a look at what manual cell counts are. In the clinical laboratory, we often rely on automated machines to do our cell counts. Not only does this make the job of the medical technologist or the clinical laboratory scientist easier, this actually produces more accurate and reliable results. However, these machines sometimes break or can malfunction. In these cases, we use now manual cell counts. These are cell counts performed using a hemocytometer or counting chambers with known dimensions. Manual cell counts also use manual dilutions made with calibrated or automated pipettes and diluents. All cell types can actually be counted this way, whether it be blood cells in the hematology section, yeast cells in the microbiology section, or even sperm cells in the clinical microscopy section. Manual cell counts involve the following steps: dilution, charging, counting, and computation. We will be tackling each of these as the lesson progresses. Let's first tackle dilution. There are three ways that you can do dilution. First, manual dilution, where you manually add accurate amounts of blood and dilution fluid in a test tube using either serologic or micropipettes. Then you can use a dilution pipette, for example, a Thoma pipette, which can be seen in the upper picture. These are glass pipettes with calibrations, so all you need to do is aspirate your blood and dilution fluid up to the different graduations in the glass pipette and mix it. Then we also have blood diluting systems such as the UnoPet or Leukocheck, and these are disposable capillary pipettes and reservoirs containing pre-measured diluting fluid. In our lesson today, we'll be focusing more on using the dilution pipette. For manual dilutions, there are two main dilutions that are usually used. First, the 1:20 dilution, which is used mainly for white blood cells. And we also have the 1:100 dilution, which is usually used for red blood cells. You should also note that dilution fluids that are used will depend on whatever type of cell is being counted. For example, white blood cells will require a hypotonic diluting fluid, such as 1% ammonium oxalate, while red blood cells will require isotonic solution, such as normal saline. And of course, if you use it on platelets, you would also want to use a hypotonic solution, again, such as 1% ammonium oxalate. In the next slides, we will also be tackling other types of dilution, for example, the 1:200 dilution, or even the 1:10 dilution. Let's take a closer look at dilution pipettes. You should note that for each type of blood cell being counted, there is a specific dilution pipette that is being used. For red blood cells, you would need an RBC Thoma pipette. You can distinguish this from its WBC counterpart using the red bead found in its bulb, the markings along the pipette which read 0.5 unit, 1 unit, and 101 units, its larger bulb, and of course the lumen, which in the RBC pipette is much smaller. There are two types of dilutions that can be done using this pipette. First, the 1:100 dilution, wherein you aspirate blood up to the 1 unit mark and dilution fluid up to the 101 unit mark. Then you also can do a 1:200 dilution, wherein you aspirate blood, this time up to the 0.5 mark, and dilution fluid again up to the 101 mark. Now, if you wanted to count WBCs, you would also need a WBC Thoma pipette. You can distinguish this from the RBC Thoma pipette using the white bead found in its bulb. The markings along the pipette also read 0.5 unit, 1 unit, and 11 units. Its bulb is also smaller, while having a larger lumen compared to the RBC Thoma pipette. There are two types of dilutions that you can do with this pipette. The first is the 1:10 dilution, wherein you aspirate blood up to the 1 unit mark and dilution fluid up to the 11 mark, and the 1:20 dilution, where you can aspirate blood up to the 0.5 mark and dilution fluid up to the 11 mark. For each blood cell type, you will also need a specific dilution fluid. For example, for RBCs, you will need isotonic fluids, such as Daisy's fluid, normal saline solution, and 0.5% sodium chloride. On the other hand, if you're doing a WBC count, you would need a hypotonic diluting fluid, such as 1% hydrochloric acid or 3% glacial acetic acid. The reason we need to do this is that for WBC counts, we need to lyse the RBCs, which greatly outnumber WBCs. This makes it easier for us to count the WBCs and provides less interference. The hypotonic diluting fluids can also be used for platelet counts. Here we have a figure showing what happens to RBCs in different tonicities of liquid. For example, in an isotonic solution, the RBCs would remain intact. Compare this to a hypertonic solution where the RBCs actually shrink or become crenated, and the hypotonic solution, which is the same solutions we use for WBC counts. In these solutions, the RBCs are lysed. Let's move on to the next step in our manual cell count, charging. This is the act of filling the counting chamber or hemocytometer with diluted blood, and you do this through capillary action. For the charging procedure, you would first need to discard 3 to 4 drops to remove excess diluting fluid. Then, holding the thumb pipette or capillary tube if you are using a manual dilution at a 45-degree angle, touch the tip to the cover slip edge where it meets the chamber floor. This should allow the fluid to fill the chamber in a steady and continuous flow. Make sure you do not overflow or undercharge the chamber. Next, transfer the counting chamber into a damp container for at least 10 minutes to allow cells to settle. You can use a piece of moist filter paper or cotton to keep the container damp. Once you have allowed your cells to settle for at least 10 minutes, you can now proceed to counting. When we count cells, we usually use a hemocytometer, or some references actually list this as a hemocytometer. This is a thick glass slide with a gridded counting chamber. The counting chamber has known dimensions, which allows us to determine a number of cells per given volume. The most common counting chamber being used is called the Levy Chamber. This is composed of 2 raised surfaces, each with a counting chamber, and these different surfaces are separated by an H-shaped moat. So here's the figure on the right-hand side of the slide, and you can clearly see the different surfaces with the counting chamber. And as you can see, the moat is shaped like letter H. This type of chamber uses an improved Neubauer ruling to count cells. Let's take a look at the dimensions of the improved Neubauer ruling. So this is actually a 3x3 millimeter counting chamber with a depth of 0.1 millimeter. Each chamber is composed of 9 1x1 millimeter squares. 4 of these squares, known as the 4 corner squares, have 16 smaller squares within them, while the central square has 25 intermediate squares, and each of these intermediate squares has a dimension of 0.2 millimeters by 0.2 millimeters, or an area of 0.4 millimeters squared. And again, each of these intermediate squares is further divided into 16 squares. So when I say an intermediate square, those are the smaller squares within the center square labeled with an R, while the 4 corner squares are, as the name suggests, found on each corner, and they are labeled here in this figure with the letter W. Now for WBC counts, we employ the 4 corner squares of the counting chamber. and these cells are counted using the low power objective of your microscope. When we count WBCs, these are the rules that we follow. First, cells touching the top and left boundaries are counted, and cells touching the right and bottom boundaries are not included. You need to count the cells in the four corner squares of each counting chamber. Once you have done that, you count the other counting chamber. So the difference between the average cells in both chambers should not be greater than 10%. For example, if you counted 40 cells in your top chamber, your lower chamber should only have at least 36 cells, or at most 44 cells. If the difference is greater than 10%, you will need to repeat your charging. Let's have a small exercise. Using the rules I just mentioned in the previous slide, count the number of WBCs in this figure. You can pause the video while you count. If you counted 12 cells, you would be correct. As you can see in the figure, we did not include the cells touching the right and bottom boundary. In this figure, you can see what WBCs look under the microscope using the low power objective. You can distinguish them from the RBCs by looking at their appearance. WBCs are larger and have nuclei, which makes them darker or speckled compared to RBCs. For RBC counts, we count the cells found in the 5 intermediate squares of the large central square, and we count these cells using the HPO, or the high power objective. If you take a look at the figure here, you can actually see that each of the intermediate cells in our central square has boundaries composed of 3 lines. So when we count RBCs, we have somewhat different rules compared to WBCs. First, cells touching the middle line of the top and left boundaries are counted, while cells touching the middle line of the right and bottom boundaries are excluded. We have to count cells in the 5 intermediate squares of the central square of each counting chamber. And once again, a difference between the average cells in the top and bottom chamber of greater than 10% means that you need to charge your hemocytometer once again, because this might be a variation in the way you are charging your hemocytometer. Alright, so we have another exercise on counting the RBCs. Using the rules I just mentioned, I want you to count the number of RBCs in this figure. You can pause while you count. If you counted 34 cells, you would be correct. As you can see here, only cells touching the middle line of the top and left boundaries were included. Meanwhile, cells touching the middle line of the right and bottom boundaries were not included. In this figure, you can see what RBCs look like under the high power objective of the microscope. You can see that they are very circular and that they have a uniform appearance without any granules at all. And as you can see in some of the RBCs, the middle part is actually lighter than the outer part. And this is because of the biconcave form or shape of the RBCs. Once you have counted all your cells, you can move on to the last step, which is computation. Using this equation, you can find the number of cells per microliter. Average cells times the dilution factor over the area counted times the depth. So average cells refer to the average of both counting chambers, and our dilution factor will depend on whatever dilution we were using in the previous steps. For example, if you used a dilution of 1:200, you would have a dilution factor of 200. And if you use a dilution factor of 1:10, you would have, if you use a dilution of 1:10, you would have a dilution factor of 10. Then of course the area, and then your depth, which is constant at 0.1 millimeters. Let's have an example. Say we were asked to make a WBC count, and we diluted our sample to 1:20 dilution, and we counted the four corner squares of each chamber of our hemocytometer. The top chamber, we had 64 cells, and in the bottom chamber, we had 66 cells. What would be the WBC count for this? I want you to solve along with me, so you can pause the video here while you solve. The answer is in the next slide. If you were able to get 3,250 cells per microliter, you are correct. Let's take a closer look on how we got this answer. First, we took the average of the two counting chambers, 64 plus 66, and we divided this by 2. Then we multiplied this average by the dilution factor, which is 20, and we divided this by the depth, which is a constant 0.1 millimeter, multiplied by the area, and since we counted 4 corner squares with each square being 1 millimeter squared, that gives us a total area of 4 millimeters squared. Once we have that, we can see that we divided 1,300 by 0.4 and we got our answer of 3,250 cells per microliter. Now this is how we express units in the conventional manner. But if you wanted to convert it into the SI units, you would just need to multiply your cells per microliter by 10 raised to the power of 6. So if we did that, we have 3,250 times 10 raised to the power of 6. This would give us a total of 3,250,000,000 cells, or 3.25 times 10 to the 9th cells per liter. Okay, this time you'll be doing an RBC count. We diluted our sample up to 1:200, and we counted the 5 intermediate squares of the middle or the central square. Our average cell count for the top chamber was 480, and our average cell count for the bottom chamber was 466. I want you to pause the video here and try to solve the RBC count along with me. If you were able to answer 4.73 times 10 to the 6th cells per microliter, you are correct. So how did we get this number? So first, we got the average of the top and bottom chambers, which is 480 plus 466. We divided that by 2, and we multiplied it by our dilution factor, which is 200. Then we divided the whole thing by our depth, which is 0.1 millimeter, times our total area, which in this case is 0.2 millimeters squared. And we got this because we counted 5 intermediate squares, and each intermediate square has an area of 0.04 millimeter squared. Given that, we now have the next equation, which reads 94,600 divided by 0.02, and this gives us the final answer of 4.73 times 10 to the 6th cells per microliter. Now this is how we express our results in the conventional method. If you wanted to find the SI unit, you would just need to multiply your cells per microliter by 10 raised to the power of 6. So here we have 4.73 times 10 to the 6th, and then we multiply that with another 10 to the 6th, and this gives us 4.73 times 10 to the 12th cells per liter. Let's have another example. We need to find the WBC count. So our sample was diluted to 1:20, and we counted the 4 corner squares. Our average cell count for the top chamber was 85, and for the bottom chamber, 70. I'll pause the slide here, and you can try to find the solution. Okay, so actually, we should not proceed to the computation in this case because the counts between our chambers differed by more than 10%. So what we need to do is repeat the charging and repeat the counting as well. Here we have the reference values for both RBC and WBC counts. For RBCs in males, our reference intervals are 4.2 to 6 times 10 to the 6th power per UL, or 10 to the 12th per liter. While for females, we have 3.8 to 5.2 times 10 to the 6th power per UL, or 10 to the 12th per liter. That is the SI unit. For WBCs, we have 3.6 to 10.6 times 10 to the 3rd power per UL, or it could be 10 to the 9th power per liter for the SI unit. Here we have a short rundown for sources of errors and comments. First, if your hemocytometer and cover slip are dirty, you should clean them first before they are used because dust and fingerprints may cause difficulty in distinguishing the cells. Next, of course, our diluting fluid should be free of contaminants. These contaminants can give us a falsely increased cell count. Now, if your cell count is very low, you may want to opt to count a greater area, up to 9 millimeters squared, to improve your accuracy. The counting chamber must also be charged properly to ensure an accurate count. An uneven flow of the diluted blood into the chamber might result in an irregular distribution of cells. So you might find that the top portion of your chamber has more cells than the bottom portion. Also, if the chamber is overfilled or underfilled, the chamber must then be cleaned and recharged. And if your chamber is filled, you have to allow your cells to settle for at least 10 minutes before counting. If you didn't do this, when you looked under the microscope, you will probably still see your cells moving, and this might give you a hard time in counting them. You might also encounter nucleated RBCs. These are present in our specimen because they are not lysed in the diluting fluid, and they are very difficult to distinguish from WBCs, which may give us a falsely increased WBC count. So if you're doing a differential count and you notice a lot of nucleated RBCs, you might need to correct your manual cell count using the following equation, which is uncorrected WBC count times 100 divided by the number of nucleated RBCs per 100 WBCs. This is from your differential count, plus 100. And then you can report your results as the corrected WBC count. And finally, the accuracy of the manual WBC count can be assessed by performing a WBC estimate on a right-stained peripheral blood smear made from the same specimen. So here are my references. Thank you very much for listening.